My dear students, I am Dr. Meera BK, working as Associate Professor in the Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. Today, I am going to talk on monoclonal antibodies, their preparation and applications. This topic is one chapter in paper 7 entitled Genetics and Biotechnology. Before getting into the subject as such, we need to know the meaning of certain terms. To begin with, antigen. Antigen is a foreign substance or a protein released from the microbes the moment they enter into our body. Then we should know what is an antigen determinant or an epitope. It is a part of antigen which reacts with antibody. Then we should know what are B lymphocytes. B lymphocytes are the one type of white blood corpuscles, and the moment the antigen enters into our body, B lymphocyte is triggered to produce two types of cells, namely memory and plasma cells. Memory cells are retained in the system as such, and ultimately it is the plasma cells which are going to produce antibodies. Then we should know what is an antibody. Antibody is a blood protein which is produced to counteract the effect of antigen. Then, what is antigen-antibody interaction? The antigen enters the system in the form of a foreign body, which is identified by our inbuilt police department, that is nothing but immune system, and as a response, the plasma cells of B lymphocytes produce antibody, and these antibodies react with antigen, and then there is going to be antigen-antibody interaction so that the effect of antigen is nullified. Then, polyclonal humoral response. When an antigen enters our body, each antigen will have so many antigen determinants or epitopes, and that many number of plasma cells are triggered to produce that many number of antibodies. Thus, we will have in our body a polyclonal humoral response. What is the polyclonal antibody response? Is often inefficient for in vitro uses because the antiserum consists of a mixture of antibodies, each antibody being specific for a particular epitope. And at the same time, also antisera contain several non-antibody proteins, and repeated doses of such preparations in therapy may result in hypersensitivity reactions, that is, allergic reactions. Thus, for most research, diagnostic, and therapeutic purposes, we prefer monoclonal antibodies rather than polyclonal antibodies. If so, what are monoclonal antibodies? Monoclonal antibodies, abbreviated as MABs, are defined as a homogeneous population of antibody molecules derived from a single epitope of antibody producing cell. These are specific for a single epitope. Thus, each B cell produces only one type of antibody specific to an epitope. Therefore, theoretically, it should be possible to create clones of a particular plasma cell derived from a particular B cell which will produce antibodies of our interest. All such antibodies would be identical to each other and will work against an epitope of antigen of our interest. Then, how these maps are produced? They are produced by what is known as hybridoma technology. Before getting into the details of technology as such, we should understand one thing, that B cells have a very short lifespan of 1 to 2 weeks, and another information, cancer cells are immortal, and therefore a fused hybrid cell is possible to produce, which will produce antibodies and proliferate indefinitely. The fused cell is called hybrid, and a cell line obtained from such a hybrid is called hybridoma. George Schuyler and Caesar Milstein developed a technique to produce hybridomas, for which they were awarded Nobel Prize in 1984. What was done? Animals like mice are immunized with an antigen of interest in order to stimulate production of B lymphocytes against the antigen. Then the mice are killed and the spleen cells are isolated from these mice immunized with antigen of interest through density gradient centrifugation. Now, we have on one side mixture of spleen cells, including some cells which produce antibody, and on the other hand, we have mutant myeloma cell line unable to grow in hat selection medium. These cells are cancerous cells, do not produce antibody, and at the same time they are mutants in the sense they are HGPRT negative. They cannot produce the enzyme hypoxanthine guanine phosphoribosyltransferase transferase because mutation has occurred not to produce this enzyme. Then the spleen cells and these mutant myeloma cells are fused in presence of PEG, that is polyethylene glycol. Normally 10% PEG is used, or instead of PEG, Sandi virus can be given. When this is done, for about every 20,000 spleen cells, one viable hybrid cell is formed, and now we have in our culture plate mixture of fused and unfused cells, and through head medium we have to select only the hybridoma cells. The mixture contains unfused B cells, hybrid cells, and unfused myeloma cells. The moment we culture these cells together in hat medium, we can select only the fused cells or the hybridomas. Then we can just select these cells, clone, so that each well contains progeny of one cell. Through that ELISA technology, that is enzyme-linked immunosorbent assay. Then we have to screen the supernatants for the presence of antibody of interest and expand the positive clones. The hybridomas producing monoclonal antibodies of our interest can be isolated and synthesized. Then, what is hat selection? I told you, whatever we get, the mixture has three types of cells. Out of these three types of cells produced after fusion, B cells remain viable for about 1 to 2 weeks and die, thus preventing the trouble of eliminating them. Then unwanted unfused myeloma cells are immortal and are capable of growing in normal tissue culture media. Therefore, a special medium called HAT medium containing hypoxanthine, aminopterin, thymidine is used to select only the hybrids. Aminopterin in HAT medium is an antifolate drug which disrupts the pathway for the production of nucleotides in the hybrid and myeloma cells. The myeloma cells are thus unable to synthesize nucleotides using thymidine and hypoxanthine present in the HAT medium as they are HGPRT negative. The hybrids, however, obtain their HGPRT normal gene from the spleen cell partner. Therefore, it can synthesize the enzyme HGPRT and thus nucleotides from thymidine and hypoxanthine in HAT medium. This slide shows the two pathways of synthesizing the nucleotides, namely the normal pathway and the salvage pathway. In the normal pathway, phosphorybasyl pyrophosphate and uridylate, and when you put aminopterin, the nucleotides cannot be synthesized. In salvage pathway, we require thymidine and HGPRT for the synthesis of nucleotides. So TK and HGPRT are enzymes required to use salvage pathways. The myeloma cells are negative for either one or both, hence are unable to use the salvage pathway. Their normal pathway is blocked by aminopterin, they die. The hybrid cells, which survive in HAT medium, contain a mixture of cells producing various types of antibodies, and therefore this makes selection important. The fused cells are cultured at a concentration at which one culture well is expected to contain only one hybridoma cell. The culture supernatant from each well in which growing cells are detected is then tested for the presence of antibody reactive with the antigen of interest. Then ELISA or RIA, radioimmunoassay techniques, are used for screening. Once the positive wells are identified, they are grown on normal tissue culture medium. Note that the clones may be subjected to another round of screening. These hybridomas produce maps of a desired specificity. Then, expansion of positive clones can be done by two methods, either by roller bottle method or they may be grown as ascitic tumors. Then the purification of maps can be done either by ion exchange chromatography or radioimmunoassay or immunoprecipitation. Then, now students, after knowing how the monoclonal antibodies are synthesized, it is the time for us to know their applicative values. They are employed as in vitro diagnostic reagents Then in pregnancy detection, detection of pathogens, measuring levels of various drugs in blood, matching histocompatibility antigens, allergy diagnostic kits. Then radio-labeled maps are used for in vivo detection of tumor antigens. This helps in early diagnosis of primary and metastatic tumors used in the detection of breast cancer. Tumor-specific maps coupled with lethal toxins are useful in the treatment of leukemia, lymphoma, etc. Then they find use for passive immunization against infectious agents. They are used in identification of blood groups. They are used in diagnosis, including ELISA test for the detection of viruses and imaging. They find application in rejection of organ transplant. Example, MAB-OKT3 in acute renal allograft rejection. They are used in the treatment of malaria. They are used as enzymes. Monoclonal antibodies used as enzymes are called enzymes. They find use in acyl transfer reaction. Then they find use in carbon-carbon single bond formation and also in breakage. MAPs have been used for purification and quantification of certain molecules present in trace amounts such as hormones, cyclic nucleotides, polypeptides, enzymes, antigens, etc. The assay is extremely sensitive and quantities as low as nano or picomolar concentrations can be detected in small volumes like 1 ml of body fluids such as plasma, urine, cerebrospinal fluid, etc. Students, till now I have briefed the synthesis of monoclonal antibodies by hybridoma technology and their various applications. Thank you.